Today, I am going to be telling you about VectorBuilder and how we can help your research with a webinar that I have titled How to Streamline Your Vector Construction. Before I dive into the presentation, I just want to quickly tell you who we are. VectorBuilder is your one-stop shop for vector solutions. When thinking about vectors, we can help you with the design of your vector. We can help you with constructing the actual vector, with the virus packaging of it. We can construct a custom library from you. We could do back recombineering and all the way to generating a stable cell line. Now, answering the question on how does VectorBuilder streamline your vector construction, I have narrowed it down to 4 main points. First is the standardized plasmid design and construction that we could provide for you. Second is the access to our encyclopedia of vector parts and expert advice from our scientists. Third is the viral packaging service with stringent quality controls. And fourth, the new services to meet your research needs. To start with, let's talk about the standardized plasmid design and construction. The concept of a standardized vector design revolves around our free Vector Design Studio. You can use our Vector Design Studio straight from your browser by visiting our website, vectorbuilder.com, where you can design your first plasmid in minutes. When you choose the system that you want to use for your plasmid, it will preload a plasmid backbone in which you can customize each of the important elements of this plasmid, such as the promoter that you want to use to express your gene, the gene or the RF that you want to express, and finally customizing the marker. Just note that this example here is for an expression plasmid, but similarly, you can design either shRNA or CRISPR plasmids using a very similar platform in which it will aid you in the design and choosing of the sequences that you need to express to achieve the plasmid that you want. Once you finish designing your vector, you will come to the vector information page. And there's a couple of elements here that I want to point out that speak about how this process is standardized. First is the vector ID. The vector ID is a unique number that is given to each plasmid that is designed on our platform. This number allows you to securely share this information with your colleagues where they can take this number and retrieve this vector information page, from our website. Second is the vector name. Each vector that is designed on our platform gets automatically named using the same convention. This means that all of the plasmids will follow the same naming nomenclature and thus keep a unified system for naming these plasmids. Next, you can also see other elements of the sequence and the parts in our vector information page, but you could always download a vector report and analyze the sequence in your software of preference. All of this information for all the plasmids that you design are kept and can be saved under your VectorBuilder account. So you will never lose sight of any of this information and keep them all under one place. So how about the actual vector construction stage? Well, there's no better way to streamline your vector construction than outsourcing it to experts. And we will use the expertise that we've had in constructing thousands of plasmids and vectors to help you get your plasmid in the fastest turnaround possible. Now, this comes very handy when you have very difficult cloning projects, as for example, when you're trying to clone a plasmid for expression in mammalian cells, but the expression of this gene is toxic to E. coli. And here are a couple of the strategies that we will use to overcome that problem. But one of my favorite ones, and one that is very efficient for very hard cloning projects, is the addition of an intron to the sequence that will prevent the expression of this gene in E. coli, but that intron will get spliced once it's expressed in the mammalian cell, and you will get similar expression. So here is some data about how that works. Without the intron, you see the expression of GFP in mammalian cells, just as normally you would expect it. But then right next to it, you see the expression with the intron, and you can see that the gene is expressed as efficiently as without the intron. Now, moving on to the next topic of the presentation, which is the access to the information on the vector components and the expert advice from our scientists. Let me show you the following. There are many options that you can use to build a vector that will work in your system. And sometimes, given that biological research uses so many different cells and animal models and different contexts for their experiments, sometimes that is not an easy decision. So what VectorBuilder has done is the following. Where throughout our website, you will see that each of our elements or most of our elements have this guide button associated to them. When you hit one of these guide buttons, it will show you information for where this system was developed. Here is the example of the plasmid for regular gene expression vector. And you can see that it will give you an overview with information of what this plasmid is useful for. And it will also link the references so you can do some in-depth digging on if this plasmid will work for you or not. This information is not only present for the plasmid system, but each of the elements that are present in our plasmids can be looked up in our encyclopedia of vector parts, and you can find the references of where this element was generated along with the sequence that is being used. So you can compare it with some other sequence that you've used in the past, and you can look up the reference to see if this will work in your system. But one of the main aspects of VectorBuilder is that we can be your research partner. And a lot of our customers just ask us and ask for advice. And we'll be honest that we might not have all the answers that you need, but we can use our expertise not only from data from the literature, but from internal data to help you make the right decision. And here is just going to show you, for example, one of the big questions that our customers ask is, will this promoter work in our system? And will it be strong enough? So we look into the data and we can see here, for example, there's the different expression from the popular promoters. You can see that they have different expression and they can vary through cell type. So we will look into the literature and compare with our internal data to see if what you're choosing will work in your system. And that is another way that we can help you streamline your vector construction. Now, let's move on to our viral packaging service with stringent quality control. One of our major expertise is in viral packaging. And this table here presents the different viral packaging services that we can offer you. In the top column, we can see that we offer at least six different viral packaging types: lentivirus, adeno-associated viruses, adenoviruses, two types of retroviruses, and baculoviruses for insect expression. This will tell you a little bit of the titer and scale that we can produce for each of these viruses. And you can see these are very high titers for each of these plasmids, guaranteeing that you will get robust infection of your cells or animal models. Uh, you can see in our website a little bit more in detail of how we produce this plasmid. This is part of our guides that is also available for viruses. But I just want to say, just want to point out the QC part of it, in which we will titrate your viral packaging preparation. We will test for bacterium, fungi, and mycoplasma present in the preparation. And we can do transduction tests to measure the ability of this virus to infect cells, and we will look for a fluorescent expression if the plasmid package contains a fluorescent marker. There are additional quality control services. These are the standard QCs, but there's additional services that are upon request, kind of showing our capabilities as a research partner for you. Now I wanted to quickly show you some data that we're generating, showcasing how you can leverage our expertise in viral systems to meet your research needs. And here, this is pertaining to how we will help you in the design of your viral vector, regarding on how you would arrange the order of the genes in a lentiviral system. Here in the top panel, you can see that we have a system in which we have the EF1A promoter expressing the M-cherry, followed by a CMB promoter expressing GFP. So this gets transcribed from the 5' to the 3' side, and you can see that the M-cherry showcased by these images, that the M-cherry shows stronger expression than the GFP. Now, if we switch the order of the cassettes, we have GFP driven by the EF1A promoter. We can see now that the GFP actually has a stronger expression than the M cherry, and this is independent of on the strength of the promoter, as these two promoters are it should express in the same with the same strength. So this kind of shows you, like, when you're designing a vector and you're unsure, hey, I want to decide this lentivirus construct and this is what I want to do. VectorBuilder, can you revise if this design is good? It will achieve strong expression of both systems. We can use our internal data to give you some expert advice on this matter and further helping you streamline your vector construction. Now, moving on to the last point of the presentation today is about the new services that we constantly develop to meet your research needs. Of course, the main reason that we're having this virtual interaction today is because of the coronavirus pandemic. And first of all, I would like to say that I wish that you and your family have been able to stay safe during these crazy times. And of course, there's been a lot of interest in doing coronavirus research, given that that is affecting our everyday lives. So, VectorBuilder, in order to help our research community, decided that we could help by providing some coronavirus research solutions. And the way we can help with this, of course, is with vectors. And to meet this demand, VectorBuilder generated several services, including coronavirus vectors. These are vectors that contains the full genome of the coronavirus COVID-19 for the generation of more viruses for either vaccine or other types of research purposes. The other main popular item is spike protein pseudotyped viruses. We have both lentiviruses and VSV viruses coded or pseudotyped with the spike protein from COVID-19. We got some cell lines, and this coronavirus cell lines are cell lines that we've engineered to express the ACE2 receptor. We do currently have some HeLAs and 293 cells expressing the ACE2 receptors and others. And finally, some animal models that we can help you get with the help of our research partners. So just quickly, I wanted to show you some data that we generated validating both our pseudotype lentiviruses and our coronavirus cell lines, and specifically our 293 AC2 expressing cell lines. In this data, we did a transduction experiment or infection experiment in which first we got wild type 293s, and we infected it with both lentivirus. This is lentivirus that doesn't have any protein in their coat to infect the cells. Then we pseudotyped it with the spike protein, and we see that in both cases we see no infection. And lastly, we have the VSV-G pseudotype lentivirus, which is our standard lentivirus and the one that is widely used for transfection. And you can see in this panel that there's a very strong infection of the cells, noted by the expression of GFP, which is what is expressed in the plasmid containing it. Now, in the second panel here, we got our 293 TAC2 expressing cells. Our bold lentivirus does not infect the cells as expected. But now we see that the pseudotype lentiviruses with the spike protein is able to recognize that ACE2 receptor and infect the cells. Lastly, as a control, we use our standard BSV pseudotype lentivirus to infect the same cells, and we see a strong, robust infection. The main thing here to note is that yes, the infection ability of the pseudotype lentivirus is not as efficient as the VSV pseudotype lentivirus. This is already a second generation pseudotype lentivirus in which we have optimized the spike protein sequence to achieve the best infection, and we're constantly developing and updating our research portfolio to provide you even more robust services or products. So if you have any questions about this, feel free to either about this or any of our services, feel free to stop by our booth or ask at the end of this presentation, where either I or one of our territory managers will happily help you out. The last thing I want to point out today is that VectorBuilder is taking all of our vector expertise and starting a GMP clinical grade vector manufacturing service in which we will be able to meet, the standard GMP parameters to produce either viral vectors or DNA vectors with a full panel of QC, flexible scale, and with a stringent quality assurance and in-person project management to meet your needs in the GMP space. So feel free to ask us about this as well. Um, and with that, I want to thank you for bearing with me today. And I want to give a shout out to our partners all around the world, both in the academia and the biotech industry alike. And please feel free to send any questions you have our way. And we're looking forward to being your research partner. Thanks again. Have a great day.